So meanwhile, good afternoon. Thank you very much for coming. I will give a short presentation about genetic methods to identify species, in particular marine species. And here you can see a couple of zooplankton organisms, pictures I took on a cruise in the Atlantic Ocean. So you have a huge biodiversity of early life forms in the zooplankton. You have fish larvae, crustaceans, you have eggs of all kinds of organisms, mollusks, whatsoever. And identification is a major problem, especially of early stages, of early life history stages. So now we come to the key. Before it was the function of biodiversity, and now it is about how to know how much biodiversity is actually there. And I will talk about DNA barcoding, so using a certain marker gene for identification, and then one method that we developed or we worked on some years ago in the project, so-called DNA chips to identify fish species. So what is DNA barcoding? I assume most of you know, but just in case somebody has not really heard about it. So we have the genome of an organism, a metazoan, for example. So here the vertebrate genome. We have the nuclear genome, and then the mitochondrial genome. Nuclear genome is really very large, but the mitochondrial genome is quite small, 16,000 base pairs, and it's very well known. And the idea of DNA barcoding is to take a certain marker gene to agree upon, and in that case it's CO1, a small fragment of the mitochondrial genome to agree upon, and to set up a database with sequences of morphologically correctly identified organisms. And once you have such a database, you can sequence this marker gene and compare it with the database, and then you can make an identification. So this was initiated 2003 in a publication by Hervet et al., who showed that CO1 could be used as a standard marker. And then in 2005, an initiative started to barcode all fish species, which are about 30,000. In the Barcode of Life initiative, the barcode of fish barcode of life. And then the first publication showing that this works for fish was then by Ward et al., working on Australian fish species, showing that CO1 is a short fragment of only 650 base pairs, and sometimes even shorter, is feasible to identify different fish species. So once you have these database and you have barcodes, so these DNA sequences, you can also use them to design probes for DNA chips or DNA microarrays. So what is a DNA microarray? You have a surface. This can, for example, be just a normal glass slide you use for microscopy. This is coated with certain chemicals, and then you can bind short fragments of DNA, usually as long as a primer used for PCR, so something like 25, 30 base pairs. In my drawing, they're a little bit shorter, but they're something like 20, 25 base pairs, and they are species specific and bind to this DNA barcoding fragment on the chip. And these fragments have been labeled in a PCR process with a fluorescent dye. And then with a microarray scanner, you can detect the signal at a certain spot where you have a species-specific probe, and then this PCR product, which is labeled with fluorescent dye, hybridizes, and then you can measure the signal, and then you know this is a certain species in the sample. So here you don't need to sequence the sample, you only hybridize it to a DNA microarray, and then you detect the signal to identify the species. That's how a low-density microarray looks like. So this is just a glass slide you use for microscopy, and these are these tiny little spots, which are the spots with the probes, which can be species-specific. So practically you could put hundreds, or on high-density microarrays, thousands or millions of spots on this microarray, and each spot can contain different probes for different species. And Some years ago, in an EU project which was called Fish and Chips, we developed microarrays for the identification of fish species. We had a consortium with partners from all European seas to cover the whole distribution range of fish species we wanted to study. And then we used also different marker genes. We used CO1. So actually, as we started with the project, the paper came out, and then we thought, okay, we'll also try this CO1 marker. Initially, we were opting for cytochrome B because this was the standard marker for fish phylogenetics and identification. And we also used 16S to check which of these three mitochondrial markers would be better to use. So from several species, up to 79 species in 16S, we sequenced 16S, cytochrome B, and CO1, and then set up a database. And based on that database, probes were designed to be spotted on the microarray for identification. So, but first of all, a brief overview about the workflow in these methods. So DNA barcoding, you start with DNA extraction, usually with single specimens, especially when you set up the database and you have your reference organisms. You make a PCR amplification, you make a usual Sanger sequencing, and then you put it in a database, and when you then have an unknown sample, you do the same process, you compare it to the database, and then you can identify it. In DNA chips, it works a bit different. You also need, first of all, this database with reference organisms and their sequences. Then you design the probes, you put them on the array, and when you want to identify specimens, you also make a DNA extraction, again of single specimens, but you can also take mixed samples. You make a PCR amplification. This is still necessary to attach the fluorescent dye. And then you hybridize it to the microarray, you scan it, you read out the signal, and then you can see at which spot, at which probe the PCR product has hybridized, and then you can identify the species. So first of all, we want to check these three different marker genes from the mitochondrial genome, how well suited they are for species identification. First of all, here the 16S neighbor joining tree. So we made a very simple neighbor joining tree. And here you can see the species, different fish species. And such and this symbol here symbolizes that you have many different specimens, and if it's very high, then you have a lot of genetic variation. Here we have also many specimens, but it's very shallow, so we don't have much variation within the species. First of all species nicely separate using 16S, except flounder and plaice. They're so closely related, 16S cannot differentiate them, so they came together here in one branch of the tree. And then we also have the gonads, Trigelidae, three different species, even from three different genera, they all cluster here together. So for very closely related species, 16S is not working, but for all other, it worked very nicely. And we did the same for cytochrome B. And here it was possible to differentiate all species. So each species and all individuals of one species that cluster nicely here in one branch of this tree. So we can identify them. And then the same for CO1, and that it was the same result. Very nice separation in different branches of the tree. So now, based on these sequence data, probes were designed, and then these probes were spotted on a microarray, and then we were testing these designed probes based on these sequences to see which marker gene gives the best results in these microarray experiments. Here a brief summary about the barcoding. So CO1 as well as cytochrome B can differentiate very closely related species. Cytochrome B has a larger so-called barcoding gap than CO1. So what is that? When you want to identify species, your ideal marker should have no variation within the species, so no intra-specific variation, or at least very, very low. But among different species, different genera, families, and So further, you should have a big variation, a big difference. This is called the barcoding gap, the difference in variation between species and genera. And in cytochrome B, it's even bigger than in CO1. So actually, cytochrome B is a little bit better suited, actually. In contrast, 16S, as I said, you cannot distinguish close related species, and there's also no barcoding gap. So when you use sequences for species identification of 16S, you run into trouble with close related species. So now we designed the probes based on the DNA sequences and spotted them on a microarray. And here you can see one of these microarrays. The red signals, these are positive signals. So the green ones we want to see if the hybridization work at all. So this is just a positive control. The red signals are the ones we want to see, where I put this box around. So we have here 2 spots with probes for zeta maxima, the turbid. And when we hybridize the 16S PCR product, which is labeled with the fluorescent dye, we get a positive signal here with the right probes. So we can say this sample is zeta maxima. Here we can see the hybridization with CO1. Here in the box, these are the probes for this species specific for CO1, but you can see we have a lot of false signals with other probes. So CO1 is not really well suited. The PCR product hybridized also on other probes that actually have not been designed for it. And in cytochrome B, again, here in the box, these are the probes that have been designed for zeta maxima. We have the signal here, but we also have some false positives elsewhere. So in summary, it turns out if you want to design probes for microarray, then actually 16S is much better suited, because here we have a clear strong signal and very few faint false positives. But if you set your threshold a little bit higher, you can filter out the false positives. But with the other 2 marker genes, we got a lot of false positives. So in this graph, it's a summary of the arrays of the probes that worked. In total, we could find 65 probes that worked, which actually was only 20% of all probes that have been designed. So this study also showed there's a big discrepancy between what you can design in silico in the computer and where you expect that it's species specific, but then you put it on an array, you make the experiment, and you see your probe is not species specific. So only 20% could be kept for 31 species. In this graph, we have here the target species that have been hybridized, here the probes, and along this diagonal is basically the match between the target species hybridized and the corresponding probe. So all your signals should be on a diagonal here in this graph. Any signal on this side or that side would be a false positive. So with these 65 probes, we got true positive signals, but what you also can see is there's a huge variation in signal strength. Some probes give a very weak signal, others give a very strong signal. And this causes problems if you want to quantify, because you not only want to know which species is in there. If you have a mixed sample, you also want to know how much. And this makes it difficult, this difference in signal strength. So that would mean you would have to calibrate each single probe with different concentrations of DNA to relate signal strength at the end to DNA concentration, which you might then transform into biomass, for example. But certainly not with multicellular organisms into abundances, but at least there could be a step perhaps to biomass. In 16S, 20 probes worked. These were 43% of the designed ones. So here, this was the best marker. In CO1, the barcoding marker, which is now generally used, only 9% of the designed probes actually worked. And in cytochrome B, it was 25%. Here you can see a few species where we successfully could design probes. There are also other methods now that came up in the past years, like next generation sequencing or nanopore sequencing, So there are also other avenues that could be tested to see which of these techniques is good to have a high throughput parallel identification in mixed samples. Because if we work all the time with single individuals, it basically is very laborious. You have a clear-cut result, usually, about the species identification, but I would say the work you have to invest is not much less than you would have to invest if you use classical taxonomy and you look under a microscope species by species or specimen by specimen and make an identification. Okay, thank you very much for your attention.